Hi, how are you? Um, so you found the room, you opened it even. That's perfect. Could you do me a favor and click on my profile and then on the bottom it says make moderator, I think. So I can invite people to speak and yeah, I'm allowed to moderate the room.
Yes. So
Can you find? So if you click on my no 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 um maybe later. So if you click on my profile, there's like different options, and one is make moderator. Oh, perfect, exactly. Thank you. Yeah, because I don't want you to be distracted with all the raising hands and chat and options like that. So thank you. How are you today?
fine. Thank you very much. Actually, thank you for helping me because I won't be able to do that. So it's my second time in these platforms. It's very difficult.
Yeah, yeah.
Thank you for the invitation again.
Yeah, sure, of course. So yeah, we'll start in 5 to 6 minutes. So in the meantime, I'll just be inviting people. I'm sharing it on Twitter, so I'll be a little bit quiet. But if you have any question, you can ask me in the meantime. Things.
Okay, thank you.
Good evening, everyone.
Oh, hi. How are you? I've been pinging people. I didn't see you're here. Um, yeah, meet Dr. Cocucci. Yeah, thanks for coming.
Hello, Dr. Cocucci. How are you?
And thank you. Good evening. Please call me Emma. It's Emmanuel is my name. So thank
Emmanuel.
you very
Okay.
much. Nice to meet you.
Nice to meet you too. Really excited to hear about your experiment.
Thank you very much. It's fantastic. I'm very excited, and it's a pleasure to have the opportunity to present here our work.
Yeah. Um, so for everyone in the audience, we're gonna start in just, a minute or so. Katerina is just, pinging people in and sharing the room. So yeah, we're gonna start soon.
Please feel free to ping people in as well and share the room so that we can have a nice discussion.
Yes, and also please tap the greenhouse on top and follow the club. Join the club, Science Society. We have a lot of rooms like this where we have speakers from all over the world come and talk about their wonderful research. Literally all walks of science, all fields, all different parts of it. So yeah, please join the club so you can be notified when we have another cool room like this one.
Thank you.
Okay. We can slowly start. And then we can always refresh the room. So, welcome everyone to the Science Society, and we are very honored to have our guest speaker here today. Meet um, Emma Cocucci. He Let me just tell you a little bit about him. He studies basic mechanism of membrane trafficking, and he's interested in how these processes deviate during cancer development compared to normal cells or healthy cells. And he did his PhD in molecular medicine at the Vita Salute San Rafael University, and he did his clinical. So he is an MD PhD. So he studied medicine and surgery at the University of Milano, and in 2004 he did the clinical pharmacology also at the University of Milano. He received several awards and and published a lot of very interesting papers. So we are very honored to have you here today and share with us your one of the newest papers. And I added the open source version of the paper to the room. So if you want to follow the discussion, please go ahead and click the link. And just the room is recorded, so we can share it with members that from Europe or other place in the world where it's currently nighttime or a time that's not convenient, so people can listen to it later. So thank you, and the stage is yours.
Thank you very much, Katrina, for the amazing introduction. It's a great pleasure to be here and present you this work. I start by just giving some brief introduction about what the work is about and the process of clathrin-mediated endocytosis. I don't know if this is something that works, and please ask me questions, interrupt me whenever you want, so that I can try to put the stage and the idea of the problem that we studied in this paper and why it we think is something very important. Um, so the idea in general is that clathrin-mediated endocytosis was discovered in 1975 by Barbara Pierce. She was able to purify clathrin coats from pig brain. This is how everything started. And what was amazing by these electron micrographs of this kind of fraction, so it was taken from homogeneity of brain, was that there was these small cages they were surrounding vesicles. So the coat is named of the coat of the clathrin. So I don't know if it's very basic, but it's because you have basically a coat that is made of clathrin around vesicle. So from there on has been a long way of thinking how something round can come out, right, from a membrane that is in general flat of have a, let's say, a curvature in the opposite direction. If you think about the curvature of the cells, if you want to do a vesicle that is internalize nutrients, you have to change the flat surface of the membrane to something that is curved. And electron microscopy gave the opportunity by, let's say, unroofing the cells, right, and looking into the cells, how they are assembled, will give the opportunity to see how clathrin is basically assembling. And what is interesting is that clathrin is a protein made of three heavy chains that assemble into a unit that is called a triskelion. So it has, let's say, three edges. And these three edges basically are able then to interact with other triskelions, generating shapes that are hexagon, pentagons, and heptagons. And this is interesting because if you think about honeycombs, right? Now, when you have hexagons, you have only a flat surface. And it's only when you insert in some structure, a structure like a pentagon, that now you get a curvature because you change basically the angle. And basically from historically, because it was possible to look in the cells and see flat and then structure that are not flat but just have a certain small curvature but are very large patches, there was this idea that you can have a complete change from flat into a curved structure by a very sudden change and redistribution of all the clathrin molecules from flat into a sphere. um, and this clearly, if happens, requires a very strong change in all the geometry and assembling of the structure and would change several properties. But you have to think that historically we didn't have the instruments that give the opportunity to have not only a very high resolution, let's say like the electron microscope, but also the tiny resolution, because the electron microscope give only snapshots of what's happening, but also giving the possibility of looking stuff while they are happening. And so is thanks to Eric Betzig, that is also Nobel Prize for the development of Storm, right? That he developed also TurfSim. Basically, is the technology that allows to go beyond the diffraction limit of a spot and gives you a more precise geometry of what is, let's say, what we can visualize. So give me 1 second to open the version of the bioarchive. So I want to see if the order, I think that the order of the figure is the same of the other paper, but I want just to be sure that we are looking at the same thing. Um, let me know if you have a question or if it's clear or if I'm saying stuff that does not make any sense for you, or if I'm boring you to hell.
No, all good. Thank you.
Okay, no, just to know because I don't hear anyone, so I don't know. Okay, so now the problem is the solution, right? The possibility to go beyond the limit and look really at what is happening in real time in a forming clathrin-coated vesicles. So if you look basically in the first figure now, there is a simulation. So here it gives you the idea of what we are looking at, right? So if you look, for example, in figure A, you can see that we have a flat lattice. So this first model, this is the model where you have a sudden change, right? If you grow this flat lattice of clathrin on the plasma membrane, then at a certain point you have to have this abrupt transition from flat to curve, and this remodeling would change completely the projection of the surface of the object over, let's say, on the surface behind, right? Instead, if you have a continuous formature of curvature, so that you insert one triskelion at a time, and once in a while you get a pentagon, right? And you make something that is spherical. To give you an idea, the typical, so here in the modeling we are using larger objects, but typical clathrin-coated vesicle can be similar to, let's say, a soccer ball, just to give you an idea. So you have in a soccer ball 60 faces, I think, something like that. 12 are always pentagons, right? But then the others are all hexagons. So if you look at the soccer ball, when you have the pentagon, then it's surrounded always by hexagon. It's the insertion also there of the pentagon that gives this change in shape. And the problem of the pentagon and hexagon is something that returns in nature, because, for example, the structure of fullerene made of carbon atoms also has the problem that only when you insert pentagons now you have the curvature. So, however, the idea is that if you then look at the idea of how, so two things. Turf seam has this property. So total internal reflection microscopy means that you have a laser beam that is completely reflected at the glass interface. When you have this reflection of the glass interface, basically you generate what is called a evanescent wave above the glass that excites basically fluorophore, right? So the propagation of this light is only for the first, let's say, 100 nanometer above the glass, okay, just to give you an idea. And this is decaying exponentially. So you excite very well fluorophore that are very near to the glass. The more you're distant, the less you can excite molecules, okay? And this is the first thing. The second thing is that is SIB, so it's structural illumination. So basically, this is, we can then go deeper in that, but the idea is that by projecting specific shapes over the image that we are watching, we can go behind the normal resolution. So now we can get the pattern of the object. And then we will come later on this so that I can show you better on an image what does it mean, okay? However, now if we look at what we expect, if we have the flat object, now we will be able, because we are looking at using a turf seam, so we can go beyond the diffraction limit, because the clathrin-coated vesicle is under the diffraction limit of light, because it's in general in the range of 100 nanometer in size, okay? You will have a change of the surface that has to reorganize from what will be the complete spread of this surface of an object that is 100 nanometer in diameter on the surface into something that is round. Instead, if we have the continuous growth, we are just growing over time. So now if you think about this from an intensity perspective, what is happening is that in one case you will have the intensity that grows a lot because we are looking at the turf seam. So when it's flat, it's always very near the glass, the molecules. So we are exciting all of them. And upon changing the shape, now you will excite less all the object, right? Instead, in the case of the constant growth, we have an increase in intensity and then an equilibrium because the molecules are also getting out from the turf field, right? And on the other side, when you project the image, so which is the surface that we are occupying with our object, this has an abrupt change because in the flat you have to have this reorganization. And so this is what is shown in F, just to give you an idea. So there, basically, you can see that the projected area abruptly change if you look at the blue, that is the flat to curve transition. And so now if you do simulation, and there we go to B, right, you can see that we have this change from the honeycomb kind of structure, that is the top blue object, so the flat to curve transition, to insert these pentagons, and then we have something that is round. And we use 70% in the simulation because it's what was postulated in the previous published work. So they were assuming that the change from flat to curve transition was around 70% of the formation of the object. In a constant curvature, instead, you will have a constant growth. Now, let's go back to the idea of turf seam, right? If I am able to look behind the diffraction limit, right? Now, if I have a flat object in turf seam and all the clathrin-coated the clathrin molecules are near the glass, I will have basically a disk, a projection, basically a disk that is completely continuous on the surface, the honeycombs, right? This would appear just like a disk in the fluorescence intensity, as is shown in the top row of C, okay? When there is then the transition, because now it becomes a dome, you will have a high intensity on the edges, while it will be empty in the side, but not because it is empty, but because the molecules that are in the top of the object are far away from the glass. And so it will appear like a donut. Is this concept clear? So in the case instead of the continuous growth of the curvature, the moment that basically we have the appearance of the donuts is because now the dome is growing and the molecules that are on the top start to be too far away from the glass to be strongly excited by the turf decaying, exponentially decaying illumination, while the side of the object still remain illuminated, and so maintain this kind of dome shape for all basically the life of the pit, that then by closing and recruiting molecules on the bottom of the pit will be filled, and this is, let's say, the one of the last picture in C. So basically, this is to give you the idea of what was the expectation. So the idea was, we have this new technology, give us the opportunity to look in an unbiased way of what is happening. So if we have a curvature transition, we should have a disk that became a donut at a certain point. If we have, and this change should be also followed by a change in the intensity, right? The abrupt change in intensity in the projection. Instead, in the case of the curvature, the dome should start to disappear early, so we should have the donuts earlier, and then the donuts should remain constant for a long time before disappearing. So this was all the postulate, right? So this is the idea of what we were looking for. And then basically what will happen is that we did images of many cell types to uncover this difference and what we could see. So now in figure 2 are just some example of some objects. So now this is the true cells. Here we're watching using different markers, either a clathrin labeled with a fluorescent protein called ruby, that basically is just a red fluorescent protein, or using the adaptor protein complex AP2 in the AP2 GFP cells, where the marker is green fluorescent protein. So what I didn't tell you is that clathrin does not do all the job alone. So clathrin, as I said, is a molecule that is made of 3 heavy chain, and that there are also 3 light chains, but the 3 heavy chain have a different binding site on their edges for adapters, and the major and most important adapter in general is the one so-called adapter protein complex 2, AP2, which is made of 4 different subunits, and basically these adapters are the ones that are able to bind both to the plasma membrane and to clathrin. So clathrin normally cannot bind to membrane. Okay, only the adapter can. And is the recruitment of adapter on the plasma membrane through by binding to lipids and to receptor, then can start the assembly of a clathrin-coated vesicle. So these are all dynamics processes. You have to think about as a cell as a very chaotic place where many interaction between protein can take place, but each one of these interaction is very weak. So nothing tends to happen, right? And when there is sufficient interaction, then something can start. So in the case, for example, of clathrin-coated vesicle, we demonstrated previously that the recruitment of 2AP2 on the plasma membrane can be clamped then by a triskelion. That will start the assembly of a clathrin-coated vesicle because gets sufficient equilibrium to have the momentum to recruit more triskelion and adapters. However, many of these structures tends to fade off, the so-called abortives, and only few can make the late stages showed, for example, in these pictures. So we have used both SUM159 that are breast cancer cell lines. So as I was saying, figure 2A, we have used the monkey cell lines, the DSC1 green monkey cell lines. We have used the COS7 cells. And in all of these, you can see that we can appreciate this growth and the appearance of these donuts kind of shape. That suggests basically that the growth was indeed of a constant curvature. But to do that, we prove it by doing analysis and an unbiased kind of approach to detect and track each single of these forming object. And basically what we did was to align this object at the first frame where donuts was appearing, and then look how the behavior was happening before and after it, right? So in figure 3, basically what you can see is that we have the projection, how the area change over time in the concern cells and how the donuts appear. And so you can see basically that the maximum area appear around the 80% of the completion, and basically shows that there is a constant curvature. And you can see again the idea of the donut is that basically because we are breaking the limit, the diffraction limit of the intensity, the diffraction limit of the light, you can see basically in B that we can discriminate the two edges of the donuts, and they are basically apart roughly 100 nanometers. So this shows basically the diameter of the vesicle. Uh, do you have questions? Do you want to?
Yeah, I have a question. I spent a good deal of time doing all atom membrane protein simulations at some point. I'm curious about is there a preference for the adaptants? Is there a preference for lipid composition in terms of polyunsaturation or fully saturated? Do you know?
Is a very fantastic question. So the very interesting. I mean, is very interesting. So the plasma membrane is rich in a phospholipid that is called phosphinositides, and the adapters binds. So the adapter AP2 binds very well to the phosphinositide PI45. So the phosphinositide PIP2, so called. So basically, as the phosphorylation on the position 4 and 5 of the inositol ring. And this is very important because during the maturation of the vesicle, actually, the composition, the lipid composition changes, and there are phosphatases that change it from PI4/5 into PI4, and these change the equilibrium
Mm.
in the structure because the AP2 will tend to detach when the vesicle basically is nearly finished, and another protein that is called auxilin can instead bind very well to the phosphinositide 4, and this auxilin brings also then with it an ATPase, HC70, which basically are able to uh, twist the clathrin triskelion in a clockwise direction and basically uncage the coat from the vesicle. So it's very important what you're bringing up. So yes, sure. So there is a huge regulatory complex kind of regulatory steps in the formation of the vesicle. Plasma membrane in general are enriched into the phosphinositide 4/5, while, for example, endosome have more the PI3, for example. And so during the transition for the 4/5 to 4 and then in 3, basically there is a change in the lipid composition that follow the change of the kind of compartment that the vesicle is going to be embedded in, right? So I think is a very good question. Thank you. Yeah. So basically, during the formation of the vesicle, however, we can consider that the most important lipid is the phosphinositide 4/5, which is the one where AP2 binds to, and also other adapter binds to that. So, for example, also the adapter protein CAM that is also mentioned in this paper, we did the experiments at the end, is also binding to the 4/5, but has also a preference for the 4 and the 5, and this in another paper we suggest that this kind of difference can be linked to the fact that the calm may be recruited also at the end of the vesicle, promoting the closure of the vesicle itself. So yeah, and again the affinity for this binding is very low, so they tends to fall apart very fast. It's like a micromolar kind of scale. So this is also important because it's pretty dynamic system, but you have many copies, right? So in one vesicle like this, you have more than 80 copies of the adaptor protein AP2, and here I think in the simulation we do is like 163 skillion. So there is a huge number of copies of the proteins that we are considering. And so here basically what happens is that then we clusterize the different vesicle according to their lifetime and therefore their size into small, medium, and large vesicle, and then we look at the projection of the area by always synchronizing them to the first frame where the donuts can be visible, basically. And you can see that by the plot we have, the plot is much more similar to what it is the area change for a constant curvature kind of plot rather than a transition. There is not this abrupt change in surface from large to small as expected from the other kind of plot. If you want it, the opposite. then fundamentally, in figure 5, we show something that is again similar, giving the idea. So you have to think. So I want to put some context here. It was very difficult for us to publish this paper because it was a period when we started to do this work that the field was extremely excited about this flat to curvature transition that clearly, if you ask us, this is not really happening. This is clearly our belief. There are groups that think completely the opposite compared to us, and it was very difficult for us to convince reviewers. So many of the experiments we have done, many are a repetition of the same concept because it was very difficult to come through and to make the people understanding how these flat to curve transition actually does not really happen, at least in the general. So let's say we cannot assume it for any kind of vesicle, but let's say every vesicle that we detected and tracked in a cell and also in the embryo of Drosophila that you can unambiguously define as a single vesicle growing alone, where there are no other vesicle very near it, so that the signal is not perturbed by any other kind of structure, is never doing a growth that is resembling the model of the flat to curvature transition. Is always much more similar and can be, let's say, suggested to be just a constant curvature object. And so here basically we demonstrate that the experiment is again to show that there is not this kind of change considering the intensity of the from what is expected from flat to curve. So again, this is what I was discussing before, right? So if you have an evanescent field, if you look at A, you are exciting very much only the proteins that are very near to the glass. The more you're distant, the less intensity you can collect from these molecules because they are not excited by the laser, right? And then when you close the vesicle near the end, you excite again many molecules because they are the ones that are very near to the glass that are closing the vesicle. And again, when you have the constant curvature, you should expect that you have you reach more a plateau because the molecules that you are adding basically are the one that you are losing on the opposite side, and then you have again an increase. And basically, again, the behavior that we get is much more similar to the constant curvature, where you have more or less an increase and then a plateau, rather than what is happening or expected from the flat to curve transition, when you should have an abrupt change also in the intensity that should decrease when you rearrange from flat to curved objects.
Well, so I'm curious.
Yeah, please.
What was the controversy with the reviewers, or what? I mean, I'm missing an appreciation of the actual controversy.
Yeah, the controversy was that basically, if you look in the papers published in the last, let's say, 2-3 years, so before our work came out, so our work remained in bioRxiv for several times. There was many reviews, for example, from other groups that were saying that the important things to be studied was how flat to curve transition was taking place and what were the molecules involved in this flat to curve transition, right? So when we were coming out saying that flat to curve transition did not really happen, right? The reviewer were just didn't really accept the data for what they were, because they were not just believing that what we were saying could be true. That basically was the controversy. So they were not believing that could be possible, that in reality was not existing the flat to curve transition and were just a model of constant curvature. That was basically the difficulty for us. So there was, let's say, like a dogma, and people were believing just a certain kind of view and were not allowing us, for example, to share our work.
Well, yeah, I mean, I've lived through that frustration before, but I want to just get a better picture of what you mean when you say there was no transition. Maybe I must have missed that. What and
So if you look here, like if you look at figure 5a, right? So if you assume that there is a flat to curve transition, right, and you have, let's say, 100 molecules when you have the assembled object, right? And when you are at 80% the of the object, you have to rearrange this flat object, right? All the molecules, basically 80 molecules around the membrane, into a curved object, right? Now, because the diameter of the object would become, let's say, 100 nanometer, the molecules on the top of the object will be far away from the glass. Okay? So flat to curve transition means that you have to break all the flat object that is made only in hexagons, right? As we discussed before. Took away this molecule, put them back adding pentagons because they have to be the same, and reach again that intensity. To do this rearrangement, this big rearrangement, basically, you would have biophysically to measure this steady increase in intensity as shown by the blue line in figure B, in panel B in figure 5, right? And at the moment of the rearrangement, you should have a dramatic drop in intensity because now you have the molecules that have to go on the top, right? Because you reassemble everything, no? And now they are not illuminated by the turf field. Do you follow me? And now you have the finishing of the vesicle.
yeah,
Instead,
I have to just keep switching back to unmute my mic from the figures. So
okay, thank you.
yeah,
No, sorry, because without seeing faces, so I'm not very familiar, so I
right.
don't know. It's very difficult.
No, but
Instead,
I'm following. I'm following. Yeah.
okay, good. Thank you very much. While in the case of the constant curvature, what happens is that you are slowly adding the molecules on the edge of the vesicle that basically is like a dome that is growing, right?
Mm-hmm.
Right.
And so you basically arrive at plateau because when you are adding molecules on the side of the dome, because the vesicle grows only on the side. This also I didn't tell you. This is my sorry, right? You see, so because when your vesicle is growing and you start with a few triskelion in the center. Basically, let's say you will assemble the first hexagon, then you will add the molecules at the edges of the vesicle, and you add more and more molecules at the edges of the structure that is growing. Right? It's growing like
Mm-hmm.
from the center to the periphery, and each molecule you add, let's say once in a while, you will add the pentagon and would increase the curvature, but you're always adding molecules at the edges because the structure is very rigid of the clathrin. As I mentioned you, when the structure is complete, you need ATP to break the cage and disassemble it. So you need a lot
Okay.
of energy, you see. So you're building it at the sides. And so what is happening is that while you're building, you add more molecules on the side, and the molecules that are on the top start to get more slowly far away from the glass, and the more they are far from the glass, the less they are excited by this evanescent field that decays exponentially from the glass surface towards the, let's say, more deep space above the glass, right? Far away from the glass.
Mm-hmm.
And so basically, this would lead to fundamentally an increase in intensity at the beginning. And when you reach the constant growth by which you add on the sides, you would have basically a plateau with a slight increase in intensity. This is what actually we saw then in cells. As you can see in the cells, for example, we see that if you align them all for the first formation of the ring, that is the time zero in figure C, you can see that according to each cell, has its little different behavior because the codes are not all the same and there is a standard deviation. This is the gray area. But you can clearly see that there is, let's say, a slow growth and then something like a plateau, right? That is what you would expect from the constant curvature. But you never see basically an abrupt change from a constant increase in intensity, then a sudden drop due to the reassemble that is necessary in a flat to curve transition.
I see. So when the vesicle is or as it's growing, and I get the part with it's growing at the edges. as it starts to reach its maximum diameter, this 100 nanometers, and the 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 membrane at that point would have a pretty sharp curvature to it, or is that where the ATP has to cleave it and pinch it off?
No, okay, no, ATP comes much later. So the point is, as you said, right? So if you think about having, let's say, a diameter of roughly 100 nanometer, right? When you are, let's say, in the range of halfway, you are 50 nanometer from the glass. So when the top of the dome is 50 nanometer from the glass, you can assume that you are exciting the fluorophore, let's say, half of the intensity, or half of the amount that you expect at the glass. So now the contribution from the top decrease, and this is the reason why we see the donuts, right?
Mm-hmm.
And you won't see the donuts basically in the flat to curve transition until the late stages, because the flat object would always be completely embedded into the evanescent field. You see?
Right.
So now, when then you build it, you have this steady increase. When then you have the vesicle that is complete, nearly at the pinching phase. So let's say when you basically see the complete circle, right? So when basically the vesicle is finished, and another protein that I didn't talk about yet, that is dynamin, is coming and is pinching the neck of the vesicle, releasing the vesicle. At that moment, the lipids can basically is the transition between a little before and when dynamin comes. The lipids basically cannot diffuse anymore through the neck of the vesicle, and so now the phosphatase are, let's say, more active because you don't have coming of new lipids, right? And so now you start to lose the concentration of the phosphonolysate for 5 towards having more 5 or 4, and now you get more 4 actually, and now you get the change in the composition. So that is very,
I
very
see.
late in the formation of the
I
vesicle.
see. And there's a whole other active process with dynamin that does the pinching.
Yeah. Dynamic. So basically, when you form a vesicle, actually is another interesting part of this process, right? So you don't need energy until the vesicle is finished. So you need the energy to pinch the vesicle because dynamin is a GTPase. So you need GTP there, and then you need ATP to uncage the clathrin triskilia. Otherwise, you don't need energy. And if you think about this, it's a very extremely smart approach for the cell, because what happens is that the cells could randomly start the formation of vesicles anywhere, right? But now the vesicle will be completely formed only when something interesting is happening. Something like, for example, capturing a receptor, and this is still something that is under debate, that is not very well cleared. So there are receptors like transferrin receptors that do not signal transduction, but seems that a vesicle that is empty, so does not have a receptor and so on, tends to fall apart. And this is the reason why I was mentioning before that there are so-called abortives that are structure that don't make to becoming a vesicle, but fall apart much earlier than what we are measuring in these papers. In this paper, we don't measure any object that we don't think that became a vesicle. So every one of these objects has a lifetime that is consistent with becoming a vesicle. Okay. I don't know if I'm clear, because
Mm-hmm.
so there are objects that fall apart in the first 5 to 20 seconds of the life of a clathrin-coated vesicle, and the true clathrin-coated vesicle, let's say, usually are longer lived than 20 to 30 seconds. Are in the range of the perfect one, let's say, is assumed to be 1 minute with 1 triskelion recruited per second, something like that. Okay, this is the general idea. So I'm talking about falling apart of object in the very, very early stages of assembly, and this can be very useful, right? Because a cell can start a vesicle anywhere, and then the only one that are able to proceed are the ones that are useful because are internalizing something. So the cell spend energy only when it's internalizing something that may be useful for itself. So I think that is a very
Interesting.
interesting.
Yes. Yes. The hexagons, about what's their length scale?
So So if you go to just yeah, so each triskelion actually is very interesting. So the triskelion is an arm, right? So it has basically is an object with the three legs, right? And from one to the next leg is roughly 40 nanometer. That, if you think about from a cell perspective, is very huge. So because a transferrin molecule, for example, that is the protein that brings iron and gets internalized by clathrin-coated vesicle, is in the range of 5 nanometer, right? So from one edge to the next of the triskelion, there is roughly 40 nanometers. So now if you look at the hexagon, uh, yeah, I would say that you have basically 40 nanometer between the two opposite edges of the hexagon, but I won't be. I think so, because basically you have to think that this is on the round surface.
Hmm. So yeah, so about a 40 nanometers, but then the whole vesicle is only 100 nanometers.
But that is the so that is the that is the yeah, it's more. So that is the diameter.
Hmm.
So is 120 diameter of a clathrin-coated vesicle of, let's say, 63 skillions. These ones that are shown here are a little larger. The point that you have to think is that the distance, we can check. Maybe the measure that I am telling you is a little larger than expected. But you have to think that this is on the surface of the sphere. It's not the diameter on the sphere, so it's longer.
Yeah.
I
Cool. So this
We can
takes
check it because, but this, the beauty of clathrin-coated vesicle, I think, is that we know so much about the structures and the done by cryo-EM and so on that it is very interesting because you can really do all these cool measurements and count the number of molecules and get all this quantitative biology and correlate it with um, structures and dynamics by imaging. That becomes very interesting because are a very good model to study how things happen. And maybe uh, I may be wrong.
well. So are they fairly regular in size? Then I guess from the geometry they should be, but
So they are different from, for sure, tissue to tissue. So, for example, the one that were purified by Barbara Pierce in the 70s were from brain, and brain generally does much smaller coats. So what happens is that no matter what, if you want to close the vesicle, you need roughly 12 pentagons. You cannot close a structure with less than 12 pentagons. This is what you need,
okay.
right? Now, the more hexagon you put, the larger will be the structure. So the smaller one that is thought to be, I think, is the so-called barrel. And so this is like a small barrel, and basically this has the minimum amount of hexagon together with the pentagon. So we are talking about, I think, 36 triskelion total. Okay. The typical one that we assume that we see in cells, and we use as an example of typical object, is in the range of 63 skillions. So it's the so-called the soccer ball, right? And this is the one that is in the range of 100 nanometer, 100 nanometer, that has basically inside a vesicle of 80. In the case of the work we have done here, because we wanted to be sure to have the possibility to clearly see the donut, right? Because this is a problem of resolution. We ran the simulations on vesicle made of 140 triskelions or 183 skillions. And these are the pictures that are shown in B and D of figure 1.
Okay.
The imaging part, we were able to discriminate the distance you see on the donuts. If you look at the figure, I think the example, for example, is the figure 3, for example, of 100 roughly nanometer from one edge to the other. So maybe are in that size or a little smaller. You see 100, 122, depends. The maximum area 122. You see the minimum we can discriminate is 100 nanometer. So these objects that we are studying here are fluctuating between 120 to 100 nanometer edge to edge. So they may be a little slightly larger than the traditional soccer ball, and they are, let's say, in agreement with the size of 143 skillion and 183 skillion shown in the figure 1. So they are a little larger. So each cell,
Mm-hmm.
let's say, can build vesicle with that are slightly larger or smaller than the typical soccer ball, and also the small one can be present. So the only way to unambiguously tell you which size of the vesicle you are working with is to have, let's say, a calibrated microscope where you can actually count the 3 skillion that you are recruiting in the vesicle.
Mm-hmm. And you say
And
that. Oh, go ahead.
sorry, go ahead.
Oh, I was just going to ask.
Please.
So the time scale is it takes about a minute for these things to form?
Traditionally, what you would say is that 1 minute is roughly the lifetime of a clathrin-coated vesicle. There are some, again, it's not it's not something that is always exactly the same. So there are some that can take a little longer, depend also on the cell line, depends on the, depend on the size fundamentally of the vesicle. You can assume that you have the recruitment of 1 trillion per second. This is roughly what is assuming. If you want to do, a approximative evaluation, but again, the average is, let's say, 45 to 60 seconds. There are objects longer lived than those. There are objects shorter lived than those. But let's say when you are looking at 60-second codes, is a code that is most likely the size of a soccer ball. So this one that are a little larger may takes a little longer. Yeah, as you can see, for example, in the case of of the figure 2, you can see that the object we show in BSC1 that there tends to be the typical one studied in the early 2000. You can see that they are in the range of 48 to 60 seconds, 48, 42. And the ones, for example, in code 7 seems to be a little longer in time, takes 120 seconds, the one that we show. So there is clearly a variability in the lifetime of the objects, and depends on many factors. But let's say, for example, this is one of the reasons why it's difficult to conceive that clathrin-coated vesicle are the only way to recycle membrane at the synaptic terminal, right? Because it will take too long to make clathrin-coated vesicle to recycle all the synaptic vesicles, right? So there are many other mechanisms involved there.
I guess I'm also curious if lipid rafts interfere with the process, or does it generally happen um, in the more fluid membranes?
No, yeah, so the lipid rafts are not involved in the formation of clathrin-coated vesicles. Usually in lipid raft you have the segregation of GPI anchored protein. These are the ones that usually are in lipid raft. And in general, high rigidity tends to result into the formation of flat objects on the plasma membrane,
Yeah,
also of
I would
clathrin.
think it would
Mhm.
interfere, but if the length scale is large enough, it may capture them. But then you'd be taking receptors or whatever was in the raft, with you. So
But the the beauty of this system is, for example, that the receptors that are captured by clathrin-coated vesicle are receptors that in general have motifs, peptide motifs that binds to the adaptor protein complex AP2, to the adaptor CAN, for example, or to clathrin itself. So in general, the protein that binds in general to the adapter. So in general, for example, in the case of AP2, the typical motif is the so-called tyrosine motif. And for my knowledge, I'm not sure that protein associated with lipid raft in general have the tyrosine motif. Instead, for example, protein associated with lipid raft usually are internalized through other mechanisms of endocytosis, for example, the so-called glyc or the one, for example, of cholera toxin, right? Cavioli or other kind of stuff like that, because they are, let's say, different kind of mechanisms. This is for my
Mm-hmm.
general knowledge, but because they are GPI anchored protein and they usually don't get in through clathrin. They're clathrin independent endocytosis. For example, I'll give you an example. EGF receptor has a tyrosine motif and is generally internalized through clathrin. However, if the concentration, the stimulation of EGF goes above a certain threshold, and now the stimulation is too high, and clathrin cannot, let's say, do the job, internalize all of the receptor, it is also internalized by clathrin-independent mechanisms. They still require dynamine for pinching, but that are different, and has been shown pretty recently that this mechanism, for example, require a reticulin, one that is a protein involved in the ER assembling or whatever. So it's a completely different mechanism. So now you can see that also protein that may be internalized through clathrin-coated vesicle may have and use other mechanism in certain kind of condition. And EGF receptor, for example, is one of those. So maybe also the opposite. So the idea that is one of the general thinking is, for example, also cholera toxin, that is a typical molecule that is internalized by non-clathrin mediated endocytosis. The idea is that if you give very low concentration of cholera toxin molecule, they may be internalized to clathrin. But now if you give many molecules that tend then to assemble or cluster, now you have internalization through pathways that are not clathrin mediated. And so there is also a problem of those that can change the way of internalization take place. I don't know if it was helpful.
No, it's fascinating. Fascinating.
And so the last thing I can share also that I think is interesting. So on the plasma membrane, we don't have only these nice clathrin-coated vesicle that forms very organized and so on. There are also still flat objects, right? Because everything started with the idea that if you look at the plasma membrane and you can see by electron microscopy flat patches of clathrin that are just hexagons. So are those clathrin-coated vesicles? No, these are plaques. We call them plaques, and they are internalized through completely different mechanisms that requires actin, for example, and are involved in integrin assembly. However, for some of these disorganized objects, has been shown that on the edges of these objects clathrin-coated vesicle can form, probably by breaking their linkage through the flat part and start to form a dome. And if you look, for example, in figure 7, here we showed that if you look at how, so you have patches, and you can see that the patches of clathrin completely correspond to the patch of AP2. So if you look at figure A, you can see that the yellowish and the cyan picture completely overlap, and also their profile if you look through these lines. So this suggests that AP2 and clathrin are together there all the time. If you do the same analysis, but in this case you use the adaptor protein calm and clathrin, now you can see that the assembly of calm is much more discrete only in some areas, right? So unfortunately, we didn't have the possibility to do 3 colors and look at all together, right? So this is clearly something that would be nice to do in the future. But now you can see that basically what happens is that the calm is much more clusterized in areas that seems to correspond to budding vesicle from this large structure, suggesting that basically it is possible that what calm does is it can influence the membrane to induce curvature. And this is interesting for 2 reasons. Number one, because it has been shown that clathrin vesicles that are much more enriched in calm tend to be smaller, because calm basically helps do doing a higher kind of curvature and then result in smaller objects. Instead, when you have a lot of AP2, now you have larger vesicles. So this goes also back to the argument you were saying before, right? Are all the clathrin-coated vesicles the same? No. No, because, for example, cells can express different amount of calm or other adapters, and the concentration of the adapters in the cell, or maybe we can even argument on the the local concentration of adapter, may drive the formation of vesicles with a larger or smaller diameter due to the change of the curvature that we can impose on the underneath membrane. And calm seems to be one of the protein involved in this. Uh, and this can be important, for example, during development. And here basically what we show is that cal most likely is recruited only in the area that are starting to give curvature to the region, and therefore it is possible that is involved in the breaking of the plaques and the generation of vesicle at the sides of these flat array that are still, let's say again, vesicle that will be growing in as a normal clathrin-coated vesicles with a constant curvature. Um, yeah, and then basically is all the story. And I don't know if you, I hope
Yeah,
was.
I was. Oh no, no, I was just gonna talk about uh I think this is very interesting, right? And I'm not gonna lie, I look at both of the models and to a degree both of them seemed plausible, like it could happen that way. But I think the constant curvature model to me personally makes a lot of sense. So but just kind of asking, what are some of the ways in which, we can apply the results of this research in probably um, other avenues of biomedical research or something like that. Like, what do you think? What are your thoughts on maybe some future projections on how we can build upon this one?
So this, is a work that is elucidating a specific molecular mechanism. I think that one of the application of understanding very well endocytosis is to being able to understand how we can build a better nanovector for drug delivery. This for sure I think is one of the avenue where being able to understand how we bend the membrane and we curve a membrane can be helpful to allow us to understand how we should design a vector for being able to be internalized efficiently. For example, this is an ongoing problem in general. So how we should design those, and for a I think that the I don't have an answer on how we can use the basic information like this. I think that this is, let's say, is an important problem to understand how a process work in general. And for example, in our case, what is can be interesting is the proportional expression of different adapters, because can lead to different kind of mechanism of signaling, right? So the presence, for example, of calm is important because it's overexpressed in many cancers. And so, from my perspective, for example, the idea that you can have a different concentration of adapter leads to a different way of doing endocytosis means that there is a different way in the cell of how you switch off signaling, right? Because the endocytosis is also involved in signaling, as I was mentioning before, for the GF receptor. So in this case, understanding which is the relation between receptor stimulation and endocytosis, and which kind of object we are going to build according to the different expression of adapters can lead to understanding how differently we can use, for example, drugs that can block signaling can be a good avenue to treat, for example, form of cancers, right? So there is all a business about understanding how we can increase the internalization, for example, of ligands such as antibody into cells through endocytosis. And again, the the number of receptors we are binding or clusterizing could affect the way the dynamic of the endocytosis can take place. And so we are working around these kind of problems, especially focuses on calm, to understand how calm is involved actually in the growth of cancer. Again, the interesting part for our perspective in the lab is the fact that the amount of calm can help forming clathrin-coated vesicle within in the increase of membrane tension, and this is important because during migration or when cells are extremely round, now the tension is maximum. So having more calm can be more helpful for uh, forming vesicle there because you have more tension, and therefore the ability to change this kind of proportion may lead to decreasing the ability of certain kind of cells of internalizing nutrients, for example, and maybe we can develop more drugs that are more cytostatic than cytotoxic, for example, for in cancer, and therefore slow down a disease rather than and target only cells that are, let's say, cancer cells, without affecting too much the rest of the cells that maybe don't need really this kind of mechanism because they are not subjected to such stress or membrane tension. I don't know if it was a good answer,
Yeah, no, it
but I
was.
hope that,
No, no, no. It makes
uh,
sense. Yeah, no, I was. Oh,
and
sorry. Go ahead.
no, no, go ahead, please, please.
Oh yeah, no, I was just saying it makes total sense, right? Because if you're looking, for instance, cancers that are specific receptor positive, right? And you're talking about the interaction between membrane receptor and vesicle formation, like things like that. It definitely does kind of consult drug formation, and thinking about how some of these particles can be be taken into the cell to react on pathways that are occurring within the cell. And when you understand exactly how the cell works, it makes it easier for you to design certain therapeutics that attack certain specific different types of cells. So, yeah, I wasn't trying to like put you in a hard spot when I asked that question. I just thought it would be nice maybe to have that on record, as part of the recording, like explaining exactly how this can be applied. But it is very important research, because the people who create drugs and who do research on diseases, they have to understand the basis of it, exactly how these mechanisms work, because that's how you can figure out a specific point in the pathway that you can attack and hence maybe inhibit something. So there are a lot of drugs that inhibit certain pathways that contribute to cancer. So for instance, the drug tamoxifen, right? So that one, it basically blocks the formation of transcriptional, it blocks precisely the transcription that occurs within the cell nucleus. So when that happens is that, yes, you have the transcription factors in there, but there's no gene expression, and that is kind of how it like helps to regulate ER positive cancer. And so if we didn't know exactly how that pathway works, we don't know what part of that pathway to attack. So it's definitely really important research, and I just want to thank you for sharing that. But yeah, thank you.
Thank you. Yeah, no, yeah. So yeah, I think that the endocytosis and the membrane trafficking is something a little overlooked in general from perspective of drug targeting. And I think that there are coming out more and more works where it's demonstrated that the modification of the expression of protein that are involved in trafficking actually have a huge impact in the cancer growth. And I have to say that one of the problems we have is that in general all our studies are done in cancer cells, right? So that is a little a contradiction because then it's difficult to know what is really the uh, normal cell behavior. But I can tell you, for example, already when you look at cells, HeLa cells, for example, have a very small amount of CAM. Uh, and instead, for example, the breast cancer SUN159 have a huge amount of CAM, and their dynamics in clathrin-coated vesicle formation is completely different, and how they internalize transferrin is has a different dynamics. And this is part of the reason why there are also controversy between the studies done in different groups, right? Because they're using completely different models that have completely different expression of proteins, and I believe that understanding how, as you were saying, right, understanding how this process works, and you have to think that forming a clathrin-coated vesicle require like something like more than 60 proteins, right, that are orchestrating and interacting one with the other. Clearly, changing the dynamics by having different concentration of each one of those can dramatically change the behavior of the cell. So I believe that this is a topic that will come up much stronger and is already coming up in the general concept of how we can target for sure cancer. That is what I'm working on, and as a general importance also in the drug delivery field at large. That I think that is relevant.
Yeah,
Yeah,
definitely.
I wanted to give a very practical example. A friend of mine has a type of cancer that ideally you don't want to touch, you just want to stop the growth because those are filled with hormones, and in that case it's serotonin in the body. So if you touch it and with some sort of drug to destroy the cancer, it can release such an amount of hormones that you will just die immediately. So the approach for those type of cancers is to ideally just completely stop the growth but leave them alone. So there are various types of these. So this would be, for example, a very practical approach to just stop the growth instead of killing the cancer.
Yeah, thank you.
I have a simple question regarding the geometry. It seems that you're artificially making a sphere. Is that correct? From the uh, hexagons, or you have a mixture of hexagons and other things. But on the paper seems to be you're showing the hexagonal skeleton. Do you have a mixture hybrid of like hexagon, maybe pentagon, something?
So the hexagon and pentagons are the, let's say, product of the assembling of objects that basically are just like that are triskelium, so are like uh, objects that have three legs. I don't know if
Yeah, you
so
mean
the
basically. Okay, go ahead.
so the basic so the problem of this paper here, is that there is not a drawing of the basic unit. The basic unit is let me see if there is because this is because the problem is that one is in its own field, and then since and here I don't have a way to show you. Yeah, it is never a picture. So basically the basic unit is called the triskelium and Catherine trischelia, and basically is an object that has three legs, let's say. And I don't know, I mean, it's funny because I'm Italian, but the symbol of this island Sicily is so called trischelia as well. Basically, the triskelion of Clathrin are called for that mythological. Basically, it's a mythological god that has the head in the center, and that has three legs that are really three legs that have basically the knees right in the middle. So has the leg, the knees, and then the foot, right? And then the triskelion of Clathrin is very similar. So I have the hub in the center, and then has the three legs that are basically built into two parts, and then an extension that is the beta hinge, that is basically the foot, and is where it binds to EP2. And they are
There's a really good picture on the Wikipedia entry for Clathrin. There's some good structural pictures.
thank you.
I have a question. So in your results that you presented are by simulation, right?
So the simulation is required. So we did the simulation to build to help the audience to understand which is the result that is expected. So we said, if you have a flat lattice, what would be the expectation using this instrumentation? And if we have a flat lattice that then does a flat to curve transition, and what is the expectation if we have a constant curvature? So we did a simulation, but then we measured the formation of the vesicle in true living cells in many different cell type and many tissues. And actually, the tissue data is the first tissue data of this kind of type ever published.
So which figure has this measured curvature data? Can you point me to a figure? Which
Sure,
figure?
sure. So the simulation is basically figure one. Figure two is an example of the data. And you can see each one is a cell type. So SUN 159 is the breast cancer cell line I was mentioning. BSC1 cells are the green monkey cell fibroblast. Nerve cells are the mouse fibroblast. And then embryo clathrin emerald is the amniocerosis. Did I see correctly?
Okay, so figure two is the measured curvature data. Is that correct?
Give me 1 second. Okay, so figure one is the simulation. Figure two is the SUN 159, BC1, COS7 cells, and embryo data. So this is true data. And then figure three is true data again. Here we show in two cell lines when we can define the dome and the separation done by the donuts, right? No, we did true measurements. This was actually this is a pretty intense uh, experiments sessions and data analysis. There is a lot of code development to analyze the data.
That's great. So this is excellent, work that you're presenting. I'm just curious, in order to, because I don't understand the flat coverage model to begin with very well. So I just so in your figure one there that
Yeah.
so one, could you, like, in your experiment just measure the so I guess my question is, I don't see a, can you repeat the figure F, like a measurement of your simulation by a real experiment as well?
So which figure do you want?
The figure 1F. So what I understand, the straight line of the intensity increasing is the until it's still flat, right? So flat on whatever substrate.
Yeah. Sorry, I got lost. Okay, so yeah, sure. Yeah. So basically, this is the one, right? So when the flat object is building up, right, you will have always the molecules near to the glass. Do you agree?
My
So if you
question,
look,
yes, my question is can you do this, repeat this simulation pattern? I mean, the result from experiments. Is that are there like experimental data show this pattern as well?
so no, we were not able to show the flat to curve because in our hands, at least, clathrin-coated vesicle never do flat to curve. The data that when we do the data that we get is much more similar to the constant curvature model.
That's what I thought, because I thought to begin with, the flat coverage model seems to be contrived in a way because you don't have such a perfect flat surface to begin with, right? So anyway, so if it's, I mean, so you're arguing against refuting the flat curvature model, and for that to happen, there's seem to be a critical point for the structure instability to kick in, right? So I saw this keyword instability rise many times. So that seems to be very significant kink or feature in the whole process that must have reflections at other, because it's a discontinuity, right? So so there's probably biological energy process you can follow. I mean, if it's a continuous, then the flat model doesn't make sense, right? Also add up to the support of your thesis.
I completely agree with you. So the problem is exactly this one. So the problem is that in the 80s they postulated this idea because they were looking at they were unroofing cells and looking at the assembled clathrin patches on the plasma membrane. And they were seeing small flat patches and then a little larger flat patches and then round patches, right? And then because I think our brain is very good in trying to make a story out of what we see. The idea was there is a flat to curve transition, and maybe the the transition and this change is where the energy kick in to change the shape of the membrane, right? So because the problem is that the grow the science come always also with the data that you can collect. So at the time there was no sufficient resolution, there was not even fluorescence, right, to look how the dynamics of formation of clathrin-coated vesicle can happen because we're the 80s. And so they started with this idea. And then basically this there was a big debate because, as you were saying, there is a strong, let's say, um, instability that is necessary to move from the completely flat to the spherical object that is very difficult to reconcile because you would have a completely destruction of the structure and then a reassembling of it that is very difficult to conceive, right? So people were saying when they did then the structural biology of clathrin, were saying it's impossible that this can take place because when the triskelion assemble, they don't disassemble so easily, ATP is necessary. So there was a lot of argument against this model already. And then lately in the 2000 I think 15 2018 came out a series of papers saying that they were using correlative microscopy, so EM and total internal reflection microscopy, claiming that this technology was now able to solve the problem because there was the dynamics and the electron microscopy. But in reality this was false because it was again a way to look at the uh, picture in EM and try to make a story of the picture looking that we were seeing without actually having the continuum measurement of how the vesicle is developing over time. And so when we discussed this paper with Chomer Corral, Dr. Corral, that is also a very good friend of mine, we got an illumination, right? We said, wow, well, with turf sim, that's the right way to go because this gives you not only the time resolution, but also the spatial resolution to really see if there is a flat object that change shape or is actually just an object that grow constantly, keeping most likely the same curvature, right? And this you can do because you have the expectation that in the constant curvature model you have a constant area that more or less you are projecting, right? And this is what is shown in the second part. And then the intensity change and reach a plateau, and then slightly increase again, as I showed you later, right? And so from that, let's say in an unbiased way, what we did was just to collect data to understand if our data was going toward one or the other option. Pretty unbiased. Let's say we come from a school because we both were, just to give you some background, right? We both were we were in the lab of Tom Kirkhausen, and he's one of the major contribution in clathrin-coated vesicle field and many structure work about how clathrin assemble and then move to microscopy and so on. So we were embedded in this kind of environment where we were a little already more towards the constant curvature model, but let's say our experiments are unbiased in the way that we had the model and we said, let's measure it and let's see what we see, right? And what we saw is something that is much more in agreement with the constant curvature than with the flat to curve transition. Now,
Yeah,
clearly you
that.
can, sorry.
Go ahead. Sorry.
No, it's fine. It's fine.
Yeah. So the I think it might be other reports that have done on various different substrate. Like say, I mean, I'm trying to uh, uh, check the, if I get the picture correct, that you have a lipid cell membrane, whatever type of membrane, and you have this particular protein, clathrin, like like single molecule, like floating around. Then somehow they self assembly and into and building up towards, if it's a flat, so-called flat uh, 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 model, that they just self-assembly as they floating on the surface of the cell. Is that correct?
Uh, I mean, they assemble by they assemble together with other molecules. So this is a very complex system that is orchestrated by the assembly of many different molecules. Adaptor protein complexes are essential because they're bridging between the plasma membrane and clathrin. What we see here is either clathrin or some adaptor protein complex like AP2 or CALM, or one or two of them together at the same time. So you have to think that there is always a limitation in our experiment design, because we, for example, had no the possibility of acquiring no more than two channels per time. So we could only look at two proteins at the same time. But this is a cell, so you have to think that it's crowded of many molecules, right? The protein is assembling together with the adaptoprotein complexes and other molecules, including receptors that are recruited in the vesicle where it is assembled. So yes, it is self-assembling, but also assembling with other units. It's a very complex process.
Uh, then the resulting vesicles, I mean, seems to be a specific. It's quite uh, definite structure with a certain number of molecules according to your figure, right? So then that indicates that somehow the unit, be it one molecule a group of molecule, the unit, uh, uh, one unit can be considered, say one vertices of the final uh, uh, crystal structure that you have. And so then the inter I mean between two units, then the tension or whatever, definitely it's much larger. I mean, shall be larger than the I mean, the tension is larger than the cell, right? So I guess I'm curious, the in order for tension to build up, there's what is the bio is it just sheer uh, uh, are there like bio like process participating in terms of energy? And I haven't had time to go into the detail of the paper.
Yeah. So yeah, I don't know. So in this case, the membrane tension, the problem of the membrane tension and how cells counteract membrane tension to do many of their, let's say, things necessary for their life is another very large field of research, right? So the idea, for example, is that if you so the membrane tension is especially maximum when you consider a cell that is completely round, right? So in the case of these cells that are growing flat and we're looking at the bottom of the cells, we expect a little higher tension on the bottom of the cells than compared to the top surface of the cell. But let's say in general, if you don't expose them to strong tension, they let's say are more or less in an equilibrium, right? They may be migrating, but fundamentally it's an equilibrium, and you cannot really measure tension. To measure tension, usually you have to use, for example, you have to use different devices, or where you can apply tension, or you can, for example, pull membrane from the cell and measure how long this membrane is pulled and and apply tension in this way. There are some papers, for example, that are interesting, where you can squeeze the cell, and in this way you can increase the tension. And in that case, you can see that basically you can block, for example, the formation of vesicle, or you can, for example, expose the cells to hyposmotic shock. This would lead water, right, running faster into the cell, and this would lead to an increase in membrane tension that would then change the dynamic of the cell. So using this approach, for example, we have demonstrated that um, if the shock is too strong, vesicle cannot form at all, for example. If the shock is mild, the vesicle can still form, and the concentration of calm, for example, in the cell can affect the capability of the cells to counteract this tension and still finish the vesicle. So let's say there is always a certain kind of tension that is there, because there is a continuous equilibrium, right, of trying to forming a vesicle. So you have to take membrane from the surface to form the vesicle. So this, in a perfect world, let's say, if the membrane is infinite, maybe there is not tension. But if in the same time you have actin cytoskeleton that keeps a shape, the cell is trying to move somewhere, there can be a local tension that can be different from one area to the cell to the other. And this may depends also how the cell is attached to the substrate where the cell is moving, right? So in these experiments, we didn't measure in the same time tension in
With
general. So I cannot comment on tension in this context, if
I
this
just
was
want
the
to
question. Otherwise, I got lost.
well, I just to add to that, what's really cool about the clathrin structures is, to think of the soccer ball, they bind along the edges of the hexagons and pentagons. They what it leaves is a fairly flat surface on the faucets for the membranes. So it's a pretty strategic approach to avoid having to induce a curvature on the membrane per se, because they get compartmentalized and segmented. It's really kind of cool.
Yeah, I think yeah, I tell you, I find this structure extremely fascinating, because this is extremely there is a lot of geometry, and there is there is a lot of information about this structure. So it's possible really to look at them and correlate what is happening in the coat in real time with the 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 electron microscopy picture and the structural information that we have about the object. So and coming back to what you're saying, also, yes, the membrane is retaining to the vesicle, right, and the vesicle is slightly smaller. So there are interesting paper where they reconstructed by cryo-EM the vesicles, and then you can see that all the coat, right? So the coat plus the membrane inside. And you can see that basically the membrane tends to be more on one side of the coat, most likely because it is more near to the portion of the clathrin-coated vesicle where it starts to grow, and then is closed down in a way that is not completely symmetrical. It's pretty interesting work. Um, and so, I mean, yeah, I think this is fascinating how they assemble.
Hi, Emmanuel. I've been listening to your presentation. Really great stuff. Um, so I have like a couple of questions. The first one is that you used Storm, and as far as I know, I think Storm can actually go to like 15 nanometers resolution. Just wondering, like, what was the limiting factor for you to obtain about sort of like 100 nanometer resolution? If there was a possibility for you to go deeper, or were you compromising the temporal resolution um, to, like, getting the kind of like lateral resolution that you would get from those iridists? Um, yeah, that would be my first question.
Thank you very much for the question. I agree with you. So our system is a SIM system. So the advantage is that we can. relatively faster build an image than compared to Storm, where you have to sample each single point, right? And so I think we had an advantage in time resolution using this system. Still, we used 6 to 9 images for each one of these plane, and this is the reason why, despite we have a good time resolution, still we have a frame, I think, every 3-6 seconds, and this clearly is limited compared to having a very detailed time resolution. So let's say, as in everything, um, there is always a trade-off, right? So the more images you would acquire in sim to get the perfect resolution, the more you will tend to bleach faster your sample, the more will be difficult to get more coat over time that forms, the less images you will get per time point, the less resolution you will get, but the longer you will do imaging. And so in our case, was a trade-off, as you said, to get the optimal or sufficient, let's say, resolution to get the exact um, shape that we require to project the areas efficiently, and the sufficient, let's say, time resolution to investigate completely the process that we were looking at. Well, we didn't also have an access to Storm, but I think that for this kind of experiment, Storm would have been, in my opinion, too slow. But I don't have a direct experience, so I don't know how fast now you can go with Storm. And the other thing is Storm in general. No, Storm, you use photoactivable fluorescent proteins, right?
Yeah, I think so. And I think STED might also
Yeah.
have like faster sampling rate as opposed to Storm, but I could be wrong. I think Palm and F-Palm might be kind of slower. I don't know how well they do with regards to resolution, but yeah, I think with Storm, you're basically trying to compromise, um, like you just said, like, the resolution, to like resolving the lateral resolution will be difficult. It might take some time when you go for smaller wavelength. So my second question was that maybe considering this kind of obstacle, was there during the study, was there at least an idea to kind of increase the vesicle side? You mentioned that calm actually, or depletion of calm, increases the vesicle size. So, in order to overcome the resolution problem, was there, did you actually consider maybe reducing calm so that you can kind of verify the sort of like the flat lattice to like the pit formation theory, because like I believe that if you have conclusive evidence, that you may not even be forced to do simulation. So I'm just curious, like if you actually try to regulate the vesicle size.
It's a very interesting and nice idea. So we didn't thought to explore this direction, um, because also we didn't want to perturbate the system. So we want just to give the measurement of how the vesicle are forming in general. And so this is the reason why we explore different cell lines, expressing only markers that, to our knowledge and our experience, do not perturbate how a clathrin-coated vesicle form normally, because we wanted to give experiment that demonstrated the model that we were looking at was the most near to reality, right? Uh, but yes, so so it's known, for example, that if you deplete calm, you get also let clathrin-coated vesicle. Depends on the cells. So if you deplete calm, you get larger vesicle in general, and this has been demonstrated. But I told you, we didn't think to change the behavior of the cells to try to investigate the system in the most kind of natural way possible. This is also the reason why we look at different cell lines, to try to give a paradigm of how the clathrin-coated vesicle form without perturbating the system too much. That was the idea. But I think is a is a good possibility to, let's say, maybe change the proportion of some adapters to push having larger vesicle, for example, or slowing down the process per se, right? That could be also useful, because if you would be having less molecule recruited per time point, maybe you could acquire more images and get a higher resolution. That are possible approaches that could be explored. I think it's a good idea. Thank you.
Gotcha. Thank you for that. And also, I guess like you kind of answered this indirectly, but besides the kind of playing around with column and other kind of or sort of like the triskelions the concentration that can basically aggregate and form these vesicles, or at least like cleave off these vesicles, is there any kind of mechanical way to kind of regulate the vesicle size? I think that you probably covered a couple of these, but I'm just wondering if there was a method or approach that can actually increase the vesicle size using mechanical forces.
I am not aware that anyone has ever been able. So the problem is that the only parameter that can tell you if the vesicle is larger is the intensity of the fluorescence of the clathrin coats that are assembling. I don't know if anyone have tried to see if so. So when I was looking, for example, at the assembling of clathrin-coated vesicle in the presence of assembling of clathrin-coated vesicle upon increase in membrane tension by hyposmotic shock. I was looking at clathrin, but in that case, my experiment were not calibrated, and the cells were transiently transfected with the clathrin marker, so I couldn't unambiguously understand if the increase of membrane tension would result into larger coats, but I could say that more calm, because the calm was instead the gene-edited protein, so was always the same per cell, that there was more calm recruited per clathrin-coated vesicle to close the vesicle. And this, I agree with you, would be a very interesting experiment that one could do to see if the change in membrane tension would lead to larger coats. And I would say that most likely this will be the case, because now it would be more difficult to close the vesicle, so now you will build a larger vesicle, because the curvature will be less high, and so it will be more easy to close even if there is a higher membrane tension. But yeah, to my knowledge, I don't know if anyone have ever tried to do it in a calibrated way, to being unambiguously able to say how much larger the coat is forming upon membrane tension increase or other mechanical stresses that I am aware of.
Yeah, thank you so much for this wonderful presentation and for all the questions. It's getting quite late, and you talked over an hour and a half, so I want to give you a chance to stop. And this was such a wonderful presentation. You explained it so well, and it was a quite technical paper for a general audience, but I think it was really great. So I appreciate it, and I appreciate that I learned that there's not really too much energy needed for that process. It's always impressive to me also that action potentials really don't require also much energy. So I always ask myself, all the brain imaging that we do based on sugar uh, physiology, I don't know, for action potentials we really don't need too much. So I always ask myself if it's the right way. But anyway, so yeah, thank you so much, and I hope you enjoyed it too, and hopefully you come back. We have more guest speaker events like tonight's coming up. So yeah, please always come back and maybe share also an update on your projects or another project. We always welcome you back. So thank you so much.
Thank you very much to everyone for giving me the opportunity of presenting. Thank you very much, Katrina, for the invitation, and for everyone for the time and the nice question. And yes, no, please, I'm not very familiar with the clubhouse, so for me is the second time that I on this platform, and I'm still not very able to orient myself. But please let me know about other presentation, and it would be fun to join and uh, listen about other research.
That was a great introduction to what I usually do in the end is tell everyone what's coming up next. So that was perfect. Thank you. Um, so yeah, tomorrow at 1 p.m. EST, we have from Oxford Dr. Cunningham talking about white dwarf accreting planetary material, his recent publication. So we have a physics room tomorrow, and then we'll have on Friday at 8 p.m. EST Dr. Mavridu coming, and she will talk about her really interesting research about breaking antimicrobial resistance. Um, yeah, so that will be also really interesting. And then on the weekend, we have Dr. Pridyarshini talking about suppressing genes across generations, and Dr. Lee from NIH coming to speak about how mitochondria act as microlenses in cone cells. And yeah, next week we'll have how neurons find their place during development and many other time crystals and how Google makes them with Charles Choi. So yeah, we'll have a lot of rooms covering different areas of science and technology. So yeah, follow the club, and hopefully I see everyone back. And thank you,
Uh,
Emmanuel, for this
yeah.
wonderful evening.
Actually,
Yeah, thanks,
can I
Emmanuel.
just ask one more question, Emmanuel, if you have time?
I just wanted to also say my appreciation, Emmanuel, and it's really fascinating that the presentation you give on the very interesting work, and especially there's a lot of I think in your paper. I mean, I see the also mentioned by Serena the Wikipedia link. I saw the stackable like structure, so that's extremely interesting. I mean, given that it's very a lot of symmetries in built in, and also from engineer perspective carrying out the experiments. It's just so challenging. So this again, thanks for sharing. And also, as because you're new to Clubhouse, so there's a function of back channel. I followed you, and I also shared a paper. Just random search a few moments ago. I thought will be relevant. I'm post that link as a question to you. The title is the thermodynamics and dynamics of the formation of spherical lipid vesicles. So apparently the constitute the units of that vesicle is lipids. So it's definitely different from your work here. But I'm just curious what's your comment on the because we're limited by time of this session. Yeah, really looking forward to a more interesting presentation of yours. Yeah, thank you. Sorry, Hanson, allow me to cut in.
Oh, no worries, no worries. Or just ask Emmanuel if yes, there was some time. But I'm okay either way. No, thank you. I really appreciate it, Emmanuel.
Molto grazie, professore Coccino. Cocucci, permesso.
Thank you very much for the paper. I will look at it. I don't, I'm not familiar with that work yet. And yeah, if someone wants to ask other question, I'm happy to answer if you want. And I would like to thank you again very much for having appreciated. And I hope that the work, and I hope that I was able to explain it enough. Thank you.
Yeah, it was quite interesting. I definitely learned a lot from it. And yeah, I always try to look at what are some of the possibilities moving forward, building upon this research, and they're endless, honestly. So it was a very interesting topic. Thank you so much, Katarina.
Yeah, just wanted to thank you again, and have a great evening or morning or day wherever you are around the world. And yeah, hopefully hear you again soon. Thank you so much.
Yes,
Bye.
thank you.
Good night, everybody.
Good
Good
night.
night.
Ciao,
Bye.
Good
ciao.
night. Bye bye. Thank you very much.